Hey guys, Corona Bry here. Just doing a little Uber Eats, and I'm looking at the coronavirus COVID-19 replication cycle. A little research over here. Trying to get my college fund together. Looks like we got some donut delivery.
More specifically COVID-19.
Donut delivery.
And we'll see once this virus gets inside particular cells of the host, how it dumps its RNA into the host cell cytoplasm, and then what that RNA does to assist in the replication cycle of the virus. So first of all, a little bit of background information about coronaviruses. This is a class of viruses, not one in particular, and they are large enveloped RNA viruses that cause severe acute respiratory syndrome, or SARS. And they have a large RNA genome.
If anybody can send some help,
that their genome size is about 30,000.
Running major low on the funds, major low on the funds, trying to work every day, get to college, super learning, active research. Donate button is in the description. Trying to stay operational. Trying to stay operational. And we're going to see a cure diseases, get to college. So if anybody can help,
and when we look at the coronavirus,
I'm full force and I need money.
It's going to have
to do it. So please, description in the link. If anybody can help, I already reached out to family, reached out to friends, reaching out to the world.
We're going to see that
moving forward.
Production of polyproteins, which are actually used to generate most of the protein machinery of the repliposome/transcriptosome. Okay? And we're also going to see synthesis of both genomic and multiple subgenomic RNA species. And we'll talk about what that means in a few minutes on one of the next slides. And so a hallmark of the coronavirus transcription process is we're going to see many subgenomic mRNAs. So these are different sized mRNAs that are created from the original RNA genome. Again, we'll look at that in more detail on one of the next slides. So here's a diagram illustrating how coronaviruses enter cells and then all the biochemical reactions that take place once inside the host cell. So notice in this particular host cell plasma membrane, we have this protein receptor called CEACAM1. This protein is actually going to be influential in bringing the virus particle into the host cell. If we look at this virus, it has these peripheral proteins around it. These are called spike proteins. And it turns out that CEACAM1 binds to these spike proteins, as you can see right here, and once bound, this protein, through conformational changes and alterations in the plasma membrane, will facilitate a sort of receptor-mediated endocytosis that pulls the virus inside the host cell. Once inside, we have uncoating, so all of these proteins will be removed, and the contents of the virus itself, which is really just this genomic RNA, will be released into the host cell cytoplasm. Now, this genomic RNA is what we call RNA positive. And I'll typically write that as RNA with this plus sign right here. That just means it's the sense strand of the RNA. But this is the genome of the coronavirus, and as I mentioned on the previous slide, it's approximately 30,000 nucleotides long. And then the host cell machinery, that is the ribosomes and so on and so forth, are going to translate this RNA strand. And they're going to translate it in a couple of open reading frames. We're actually going to look at that on the next couple of slides. So first of all, we have the coronavirus RNA sense strand. This is the genomic RNA. And we're going to get translation via the host cell machinery. And ultimately what we're going to get are two polyproteins. These are very large proteins that come from this RNA. And those are polyprotein 1A and polyprotein 1AB. Now, as I mentioned on the very first slide here, something interesting in coronavirus replication is that during this translation to get these polyproteins, we're going to see some frame shifting. And so the way that we get these two polyproteins right here is via frame shifting during translation. Okay? So you can see here we have an open reading frame 1A, and then we have over here open reading frame 1B. And this section between this point right here and this point, this is what's going to give us these polyproteins, and sometimes they're referred to as replicase polyproteins, because the proteins that come from these polyproteins are going to be involved in replication and further transcription. Okay? And so what this allows is, as translation is occurring, we can clearly get through this open reading frame 1